Hi, it's Mr. Buckingham, and this video is on enzymes. So cow's milk has a lot of lactose, and lactose is going to be the simple sugar. It's a disaccharide, and we are able to break lactose down into two smaller subunits because we have the enzyme lactase. And remember, the suffix ase means that it's going to be an enzyme, and lactase is pretty large. Sometimes people don't have lactase or lactase doesn't work. In this video, we'll talk about what happens when that does not work. In this video, we'll talk about enzymes and their structure, and they're going to be proteins. They will have what's called an active site, and that active site is going to help with the building or with the breaking apart of certain molecules. And we can call that breaking or building reactions for biology, and the catalyzation is going to be the starting of a reaction of some sort. And that molecule that we are catalyzing the reaction for is going to be called the substrate. So the example of lactose, that's going to be the substrate. Lactase is going to be the enzyme. Enzymes need to be activated sometimes, and we call those molecules that help with the activation cofactors and coenzymes. Enzymes can also be inhibited. We can slow down or stop the process of enzymes, and there are two types: non-competitive, and if it's non-competitive molecule, it will bond with the allosteric site on the enzyme. They can also be competitive molecules, and they will compete for the actual active site and block the substrate that's trying to be changed. So the substrate, in this instance lactose, will have a certain shape, and the certain shape will be able to fit into the enzyme. And we're going to call the parts that it can fit into the active site, and it's going to fit like a lock and key. If it changes shape at all, it's not going to be able to be catalyzed. The reaction will not proceed. So when they actually do fit up, it's going to be called induced fit. And then the enzyme can work its magic, and then it will separate into its two parts. Lactose example, sometimes it can be built, but glucose and galactose will separate from each other because the enzyme lactase was able to break them apart. If people don't have lactase and they're not able to separate glucose from galactose, then that's when people are lactose intolerant, and that's when they have a lot of indigestion and it's really bad. However, when people take the Lactaid medication, they can actually put some of that lactase enzyme into their body. And it's not the most healthy for you, but it's a way that people can eat ice cream because everyone loves ice cream, and it would be really bad if you could not eat ice cream and you really wanted to. So Lactaid is an option, and it's just a supplement that has lactase enzyme in it. Some enzymes need to be activated, and there are two different types of molecules that can help with that: coenzymes and cofactors. Coenzymes are going to be what's called carrier molecules. And they, for instance, could carry electrons and deposit the electrons to an enzyme, and the enzyme is able then to perform a certain function. Cofactors, on the other hand, are going to be able to participate in the actual reaction, and they are going to help with the catalysis of the reaction. Catalysis, I hope I spelled that right. And they can speed up the reaction if they need to. And we can find these molecules in our diet, and they can be either vitamins or minerals. So this is why you need a full balanced diet, and if you don't, then sometimes our enzymes won't be activated like they should. So the vitamins can be vitamin B, vitamin C. Orange is a good vitamin C carrier. Scurvy is a good example of when people do not get enough vitamin and minerals, and they're not going to have those cofactors or coenzymes. And vitamin C plays a huge role in that, and I know that you have learned a little bit about pirates and when they got scurvy because they're not able to get enough of the fresh fruit or produce with them when they are traveling the seas. So vitamin C helps with the production of collagen, and that absence of collagen is why we get the sunken eyes, loss of teeth, and the pale skin. So what actually is happening, and I go beyond a little bit with the biology of this that you need to know, but vitamin C is going to act as a cofactor. So it's actually going to help this enzyme, lysyl hydroxylase, to become more of an active enzyme. And so it will go travel over to the enzyme, and it will help with the connection of an OH group with lysine, and lysine being an amino acid. And when that happens, and they all connect, and it's an active enzyme, then lysine will be able to go over and help the production of collagen. And then it's not going to be a sad pirate, but a happy pirate. We can also shut down the processes of enzymes, and that can be with non-competitive molecules or competitive molecules. Non-competitive molecules are going to bind, bind not in the active site, so elsewhere, and the competitive is going to bind in active site. So it's going to compete for the spot with the substrate. Substrate will want to normally bind into the enzyme active site. However, the molecule that's going to inhibit is going to be the competitive inhibitor, and it's going to take up the active site. So now the substrate can't get in at all. If we want non-competitive inhibition, the molecule will bind to what's called an allosteric site. That's not in the active site. And when it bonds there, it could actually change the shape of the active site. And it could be pretty pronounced. So when the substrate actually tries to fit in there, it actually won't go into induced fit. Another type of inhibition that could happen is where the molecule, the non-competitive inhibitor, will actually have this structure that physically blocks the active site. So this substrate is no longer able to fit into the active site. Enzymes can denature, and denature is where the enzyme changes shape, and we call that conformational change. And what's happening is that a lot of the bonding processes in the protein are going to change or are broken. That means that the enzyme is no longer able to work. And there are different ways that that can happen. One is really high temperatures. That can change a lot of the bonding structures. The next one can be pH. So our stomach acid is a really low pH, and that can denature some of the enzymes. And the last one, salinity, can greatly, so extra salt can greatly change the shape of an enzyme. Our bodies have different enzymes that have different optimal pHs, and they will work the best in a different pH. So this blue curve will work the best when it's at a really low pH. Let's say this is about 2 to 3. That could be our stomach acid. So some enzyme could be in our stomach hanging out and being happy as a clam, and it is able to function in there. That same enzyme will not work in a pH of around 7. So if we were able to take that enzyme and put it elsewhere in our body that was more of a pH of 7, it will not work. Reversely, we can have this green one. It might work better in more of an alkaline area in our bodies or in some other solution. All right, go back and see if you can fill in the missing text boxes, and hope that helped. Thanks.